Okay, so maybe to just kick off the session, I'd actually like to have my co-moderator, Karen, maybe talk a little bit about treatment landscape in I&I right now and some of the limitations.
Great, thank you, Susan. It's very nice to be here. This is an exciting panel. This is what I do. I'm a rheumatologist, and I see mainly patients with lupus and also rheumatoid arthritis. And so when we talk about I&I indications, I think we really haven't had precision medicine, and we're getting there, so it's very exciting to think about the concepts that they already have in oncology and other diseases for our diseases. So what are our diseases? The autoimmune diseases in general affect probably 1 in 10 people, have substantial morbidity and mortality. I see rheumatoid arthritis and lupus, and psoriatic arthritis and systemic vasculitides, and then beyond that there's inflammatory bowel disease, Crohn's disease, ulcerative colitis, multiple sclerosis. There really are hundreds of these autoimmune diseases, but then we also talk about inflammatory and allergic diseases. So there are many, many indications. So it's very exciting. Right now, to set the landscape for this, where we're talking about novel therapies, what do we do for treating these patients? Well, we have some clinical guidelines, but they're not really precision approaches. We have new medications approved, which is very exciting, but we try them kind of in a stepwise fashion. You know, we try this for everybody. If it doesn't work, we move on to the next one for everybody, and if that doesn't work, we move on to another one. And we're always balancing, you know, risks and benefits, but we're not really thinking about the particular pathways involved for that particular patient in front of us. So that's what I think is really exciting about these novel therapies and novel precision approaches that we might be bringing to our field.
So with that, why don't we have each of our panelists, starting with Tarek, talk about their company and their approach to treating I&I.
Sure, so I will start. Thank you very much to the World Medical Innovation Forum for inviting me to your World Forum. So I'm Tarek Kassim, the CEO of Celsius Therapeutics. I'm a medical doctor by training, but I've been on the business side my entire career. And I joined Celsius in 2019 as its first full-time CEO. What we do at Celsius is we use a platform approach that involves human tissue samples and single-cell RNA sequencing to better understand the cellular landscape of disease. Using this approach, we've identified a target in which we have very high confidence and we're very excited about called TREM1, which is an amplifier of inflammation that is seen to have a very prominent role in inflammatory bowel disease and also other inflammatory conditions. And so we have an antibody directed towards that target called CEL383 that is on the brink of its first-in-human testing. And so we're planning on advancing that program in ulcerative colitis and Crohn's disease. Notably, and going to the point of the point of the panel, we've also identified biomarkers of TREM1 pathway dependence that we are going to analyze retrospectively out of our phase 2 study. And then if they do predict enhanced responsiveness to CEL383, we can consider using those as prospective patient selection in subsequent studies. So new target, new approach to patient selection.
Great. Jody, why don't you tell us about
Great. Hi, I'm Jody Morrison. I'm currently the CEO of Q32 Bio. So Q32 Bio is focused at its heart, really in its platform of building new complement therapeutics that actually direct and hone to the tissue itself. So avoiding the systemic approach, going directly to the tissue, using kind of a fusion protein style approach in both antibodies and nanobodies. Our lead compound is now through phase 1 and entering into phase 2 later this year is our hope. We're raising capital for that as we speak, and the goal there really is going to be looking across 4 different indications, targeting this approach, right, for complement in a directed way. So we think about it as sort of the systemic approaches that are out there are sort of draining the ocean of the leaky boat. We're going directly and repairing the boat itself. That's the approach that we're taking. We also have a bifunctional antibody that's in combination with Horizon right now and an option. That's an IL-7 TSLP asset that's in atopic derm in a second phase 2 trial right now. So we're taking precision approaches across our programs and looking at the data that we're collecting there, looking for ways to enhance biomarkers as well as to identify patient stratification approaches for the future.
Thank you.
Perfect. Hi, I'm Jason, CEO and co-founder of Endpoint Health. Thank you for inviting me to the panel. Serial entrepreneur, so I've been building and running healthcare companies for a while. Endpoint Health is a precision immunology company, and when we think about precision, I appreciate kind of the point of the panel. You know, what do we mean by that? Can you pick out the patients that should get a therapy? Can you get the therapy to the place that it should get and hopefully not elsewhere? And can you have it function when it needs to function but not otherwise? That would kind of be the holy grail if you could stitch those pieces together. So at Endpoint Health, we're taking a shot at it. So we have an AI-driven precision medicine platform. What we do is really we're working on redefining autoimmune diseases. If you talk to folks, these are very, very heterogeneous illnesses, and we've discovered a particular way of identifying biological subgroups that seem to be present and consistent across many different autoimmune and inflammatory diseases, which makes a bit of sense in that you have one immune system, but you may have many different ways it could go wrong. We use that platform to stratify patients, and we've been able to back test and see that patients in these groups definitely respond in different ways to existing therapies, and we've used that data to pick out what we think may be a better precision therapy, which is our lead asset. It's a monoclonal antibody targeting tissue factor, which is a very interesting target, a different way of getting to a bit of extra precision in that it's a dual mechanism of action antibody that inhibits both integrins and cytokines, but importantly also is only really expressed and available when a patient is under pathological condition. So a different approach, but very important. I love the stacks of precision here that we're trying to get the right drug to the right patient when they need it. So happy to be here.
Great, thanks.
So building on this idea of what is precision, I just wanted to maybe start off with Tarek again. What does it mean to rebalance the immune system, and why are current approaches maybe failing to do that in some patients?
So that's a big question. The immune system in the context of inflammatory disease, and I'm thinking particularly of inflammatory bowel disease, you think about what's going on in a patient with IBD. There is a derangement of the cytokine, chemokine, and cellular milieu in the tissue of these patients. There's and part of it that makes it so complicated is all of those different elements talk to each other. This soup is incredibly difficult to disassemble. And so the current therapies that we have and the current therapeutic approaches, I think, are very, very effective at addressing certain threads of this tangle. I think what all of us are trying to do in some way or another is find a therapeutic approach that addresses multiple aspects of this multidimensional problem. So for me, as I think about rebalancing the immune system, I think less about the whack-a-mole of hitting like one cytokine, but rather what can we do to address a number of inflammatory mediators, a number of cell types, and hopefully be able to prevent the continuous cycle of dysbiosis, inflammatory signaling, repeat, recycle.
This question may be for Jason. Are precision approaches more amenable in some I&I indications than in others?
So, there's a lot of different ways to answer it. I think one of them clearly, first on all of our minds, is going to be, well, where do you have the biggest unmet medical need? And that's something we talk quite a bit about. So if you're going to take a precision approach, what are you really trying to do? You're trying to amplify efficacy. You want to not give therapy to folks that won't respond, give it to the folks that will, because there's two very important things there. One, you want to make sure you get the right treatment to the patients that will respond. But currently, if everyone gets a treatment, then you're blocking the next therapy from coming in. So I think, one of the things you're going to need is an area where you've had some efficacy ceilings. I think areas like lupus, areas like IBD, and there's many, many other autoimmune and inflammatory disorders that have that. I think also a lot of us will make decisions based on our technology and our platforms. We'll choose places where it's a little bit quicker to figure out how to find the right patient or understand what particular target you need to address in a particular patient group. In IBD, you have tissue available, and others you don't. But I think, it's really a layer. It's just like indication selection. It's going to be a layer of questions. And I think a big one I'd be interested almost in the moderator's point of view. It's going to be a question of which clinical indications are there, interest from the clinical community. Where are they looking for something better for their patients? Everyone is, but some places are faster to adopt. We all look at the oncology panels and see where they've gone. And you have to wonder who's ready to move into that. So it's a really complex question, but a good one.
Yeah, great. So following up on all of your intros and your 3 different novel targets, I think I have a lot of questions. So in clinical medicine, we use clinical phenotyping diagnoses. But do you envision that any of your novel targets or any other novel targets will be useful for phenotyping people differently and precision approaches across our diagnoses, possibly?
Yeah, I'm happy to
whoever.
go. Yeah, I think we certainly do from the perspective of Q32. We think that's where we're headed. How we get there from a development path is a different question, right? How we think about working with the regulators, working with the clinicians, and driving our programs forward. Are these companion diagnostics approaches, or are these sort of enhanced data collection that will allow us to sort of type and focus our patients, stratify our clinical programs? That's how we think about it. Q32 hones to the C3d that co-locates with the convertases within the tissue that's inflamed in the setting of autoimmune conditions. So the concept, we've looked about 80% of the histology we've looked at in a lot of the core diseases in renal as an indication, kind of area show this co-location. So do we need to pick those patients and just do that 80% and build a companion diagnostic? Do we need to stratify that 80% across our clinical programs? That's how we have to think about it. There's a lot of regulatory components to that, but the agency is very open in the discussions we've had with FDA in those conversations and thinking about how to collaborate on that. So I think we're headed there, but it's going to take a while.
Yeah. So just one more question. I think we're going to come back to the companion diagnostics question as well. But I think when we're talking about this idea that like of immune homeostasis and rebalancing, do you envision different novel targets being used together in the future? If we are trying to downregulate certain pathways, are we upregulating others, or trying not to overshoot to over-immunosuppress patients? Will we get to that level of precision medicine for our patients?
Well, I think that we're already starting to go down that path, but without the precision. And so you can see the example there in combination therapy in IBD, right? There was a J&J study called the Vega study that mixed a TNF inhibitor and a P19 inhibitor, showed increased efficacy. Yes, as expected, a higher rate of adverse events. Combination therapy in IBD is a very, very hot topic as a way to do what you just described, hit multiple pathways simultaneously. The problem is, even there, you're dealing with a minority of patients who are responding. And we don't know which one, we don't know which ones are dependent on pathway A and which ones are dependent on pathway B and which ones are dependent on the combination of pathway A and B. So, and then we're not even talking about the problem of getting that reimbursed. So while I do think that mixes of new, we'll say advanced therapies can be very valuable, I think that what will likely do the most benefit for patients and be most practical is having a single therapy and being able to identify those patients that are particularly reliant on that pathway. Again, stealing a page from the oncology playbook.
I completely agree, and I think it's the combination of where you can use these depends on the knowledge around the disease itself. Do we actually understand the nodes that are causing the disease, what's driving the disease, coupled with it, do we have the ability to really think about our trial hitting that in the right way in a context of a clinical trial, right? Can we address that within a clinical trial is a key question. I think there are examples of that, but I think we have a ways to go. And as for the combination, I think the goal would be to get away from it, right? If we can find exactly the precise approach that's going to address it, that's the goal. A lot of the therapies we're using now, look at systemic complement inhibitors, are broad stroke, just shutting down the systemic complement. That's not necessarily a good thing. And so then you've got a whack-a-mole infection and you've got to do different things along the way. The more we can move towards targeted approaches, the more we can get away from those combinations that are actually addressing off-target effects. And that's, we don't need that in these patients. They're sick enough. So I think we're trying to get towards that, and there'll be some backflow for that as we move forward on the precision side.
Yeah, I think it's a good question, and I think the answer is the science is going to drive the answer, because at the end of the day, if we end up seeing that there are different subgroups of patients, that they have an array of things that need to be solved, well, if that requires two therapies, then that's where the science would drive us. I think, a lot of the push to look at combination therapies now is, there's a bit of desperation in these areas where you haven't gotten a big bump in efficacy with safety, in that it's a shot to make something better. But you can see in very, very complex illnesses, infections. So combination therapy, drug resistance inhibitors and antibiotics in an infection, that's a standard thing, because you're solving two problems. But it means you have to know, to your point, you have to have the data first. You have to be able to understand these patients. It can't just be a shot in the dark, because you're adding risk and gambling on efficacy rather than making a clinically driven choice. And I think with better precision approaches, we'll get there, but we'll get there with a very rational approach.
Thanks.
So diving a little bit deeper on biomarkers, just maybe to Jason first, where are we on validating biomarkers, and what are some of the new biomarkers that you've maybe identified?
Yeah, so two parts. So I guess where are we at validating biomarkers? So each of us have a very different approach. I'll say we're the we. So I think we're all in different stages. So one of the approaches that we take in particular is we develop biomarkers. We develop relatively sophisticated diagnostic tests that stratify patients, and we can back test them today. So we already have validation from both observational studies, patient registries, and clinical trial data that the way we look at patients drives treatment response. We can predict therapy response now. That's much, much different than saying the biomarkers or tests that we have picks out responders to the drug we're developing. That's obviously something that has to come with a robust data package. You tend to get the first peek of that after a phase 2, or at least a reasonable bolus of patients. I think that we're probably, everyone has seen Prometheus. They had a relatively good readout. Everyone can see that. I think Pfizer, now Roivant, had a biomarker-driven strategy for their TL1A. Both of those, had a biomarker approach. There was good results with it. So we've already seen that it can work at least at a proof of concept stage. I think the question is, who's going to be the first to get an approved therapy with an enhanced claim? And I think we're not terribly far away. There's a lot of smaller organizations that are pushing it, but there's a lot of folks in the audience that are also thinking about it for their pipeline. So I think you're in 2 to 5 years, maybe sooner. It depends. I don't know.
Yeah, I think it depends on the indications, right? And it depends on how you're using the biomarker. We have seen approvals, right? In renal disease, we've seen approvals on proteinuria endpoints as an example, right? The agency has embraced that as a surrogate endpoint because there's data that shows that actually predicts how patients do based on the reduction in protein in the urine. And so I think we are seeing the agency leaning in on that. We need more data to continue to drive it, and whether we'll get to the point where we use that in a stratification way to self-select patients for clinical programs from a biomarker standpoint is a different point. I think that's where I think we really need to think about biomarkers in different ways. But I do think we're making significant advancements as we think about complement, as we think about renal indications at large around biomarkers.
Yeah, well, thanks for that clarification. I think it's true. Biomarkers can mean biomarkers for patient stratification, for targeting our medications towards them, or for outcomes.
Exactly.
And for endpoints in clinical trials. And we need biomarkers for both.
We do.
And you had brought up, related to that, about companion diagnostics and developing tests that might, and you mentioned Prometheus, et cetera. So do you think that's the way we're going? And do we need further patient stratification? And then another question, how will that help or hinder the regulatory process if we have a companion diagnostic and then medication may only be approvable for, approved for this subset of patients?
Who wants to go first?
Yeah,
Well,
whoever
so
wants to take that.
I'll tell you, I think we all have a lot of thoughts on this topic.
Yes, we do.
As much as we all have oncology envy in terms of precision medicine in I&I disease, I think that the paradigm here is likely to be different. And it's not about companion diagnostics in the sense that there might be a one-to-one relationship between genetic derangement or some kind of cellular phenotype and responsiveness to the drug. I think that when you're dealing with the complexities of the immune system, we are naturally going to be moving more in the direction of what we'll call complementary diagnostics, where the presence or absence of a particular biomarker predicts enhanced but not guaranteed responsiveness to the therapeutic agent. That's what we saw with Prometheus. That's our own expectation. So for us, we have TREM1 as a target on monocytes and neutrophils, and it sort of, without preempting the patent we're about to file later this week, kind of leaves footprints. And we're going to be detecting those footprints in blood and stool. So will that be a one-to-one relationship between responsiveness to our drug and the biomarker? I mean, odds are probably not, but what we'd like to be able to do is see enhanced responsiveness for those patients that have the footprints. And I think that's a paradigm that we all need to recognize and get behind, because that's actually practical and achievable.
Yeah, so I agree. I think companion diagnostics are a phenomenal way to build and select patients long term, and I would love to see us get there. I've got the scars of running a companion diagnostic program in phase 3 in tandem with an oncology asset that didn't work out the way I wanted to, so it's a little tricky to think about how we take that on, because effectively, if you do as a company, right, you're running 2 development programs. You're developing the companion diagnostic and thinking about how to scale and eventually launch that, and you're trying to get the development asset around the table, over the table too. So those are really hard things to do at the same time, and I think very hard for small biotech to take on. And so I agree. I think I'm thinking more about it from an endpoint, from a stratification standpoint, and I think there's a lot of room for growth there, right? I don't think, we'll get to this, I think, in another question, but I don't think it's a technology limitation. I think it's utilizing the data we're getting across urine, blood, fecal matter, looking at histology in diseases like lupus as an example, right? I think there's ways we can utilize things that are already done within these patients and actually drive more data that's going to give us more clarification in our development programs long term.
Oh, that's exciting.
Jody, thank you for that segue. My next question was, are we limited by technology, or is this more about clinical validation? Why don't we start off with you?
Well, I think I made my position clear. I don't think we're limited by technology. I don't think there's some technology over the hurdle we're waiting on. I think we actually have the tools. We need to utilize the data and perhaps expand the data sets we're using and gaining from these patients, both at diagnosis and along the treatment paradigm. But I think, to me, it's really about using the data we have across these different ways we're collecting data from patients right now in a lot of the autoimmune conditions and inflammation conditions.
Yeah, I completely agree. We are in a golden age of being able to capture information about patients, even preclinical data, in a way that almost just baffles the mind. And I think how we analyze, how we look at that information, if we want to pick the polar opposite of the negative AI news that can come out and find where's the best place to apply some tools like that, it's in analyzing and understanding the very rich data that's actually relatively inexpensive to capture in clinical development, in patient registries today. It's what do you do with it? Are you asking the right questions? I do think it's worth even mentioning that we have to be really careful when there can be excitement around new modalities, new technologies that come out, because as we've mentioned, we have to figure out how do we turn that into a physical test run by a real person or a lab everywhere on the planet at launch in perfect synchrony with our drug. And that can either run on existing systems and existing infrastructure, or if you really want to bring a new technology in, it can almost harm the industry more than it could help. It's great for discovery, but how you pick your tools brings people stress, or it brings a much smoother, I won't say a smoother development program.
Yeah, I'm not sure if I agree that it would harm the industry, but I think it can harm individual companies that are trying to do it, right? Because those are different skill sets that our companies are not necessarily built to do, right? Scaling and building a lab test, be it a circulating tumor cell approach, which is what we were doing, or be it, a urine biomarker, scaling that and getting it out is not something that a small biotech company, typical size of 50 people, is, prepared to do. We don't have the lab technology expertise within our organizations. We have development expertise. That's what we're really good at. So I think we have to piggyback on the work that's being done and utilize that, I think, for the time being. Because I don't think we don't need special technology in these companies to do it. It's really just using the data that's out there in a more precise and appropriate way.
Thanks. I think, Jason, you touched on AI. And do you think that using your AI platform, or this is for anybody, will we think about, I led off talking about how we think about these clinical diseases and phenotypes based on manifestations and symptoms. Do you think that these precision approaches will lead to us thinking about these diseases differently and thinking about I&I indications differently based on the biologic pathways involved?
Yeah, the very hard answer is yes. I mean, this is, if you had to pick an area of medicine that is kind of almost designed for using older AI tools, newer AI tools, this is it. And what really defines that? And Tariq said it to begin with. This is a very complex set of diseases. You're talking about the immune system, and even the point of the talk, balancing the immune system. You're trying to understand a dynamic system that interacts with every part of your body at every stage of your life. You have feedback loops. You just have terabytes of data that could be generated with every sample. You just described a place where a individual or even a room full of mathematicians would cry themselves to sleep, but where you have a beautiful problem for a thoughtful AI approach. It still has to be, you don't just sprinkle on technology and expect a result to come out, but it is a phenomenal tool. It's what we use, so I am a bit biased, but we've seen fantastic results. And case in point, our core technology, like the way we measure information about patients, is actually purposely quite robust, it's not the newest modality in the world, but the way we look at data with AI is newer. And that has led to the success we've seen, and there's a lot of different ways to do it.
I would just say this relates to a question you asked earlier about, do we need more patient stratification clinically to be able to identify precision or identify subpopulations? Or, as you're asking now, is this going to be something that's more driven by increased biologic understanding of disease? I would just say that we've tried for decades and decades to clinically subdivide many of these conditions using different different slices and dices, and we still haven't been able to pinpoint, hey, this is what a patient who's going to respond to an anti-TNF looks like. So I think naturally we have to go down the road of better understanding highly complex biology, and the computational tools that you described, I think, are a prerequisite for doing that, because there are way too many variables for anybody to start sifting through this by themselves.
Yeah, well, I couldn't agree more about the years we've spent trying to dissect the different subpopulations of the different autoimmune diseases. And the feeling is now that probably some people with rheumatoid arthritis have more pathways that are similar to lupus or similar to IBD than others. So I think that will evolve, and so it's really exciting to know those tools are out there, et cetera. Are there any questions? No.
Just as a reminder, you can put questions in the app, and we will ask the panelists. Maybe just segueing to regulatory questions. We just had former FDA Commissioner Scott Gottlieb, and yesterday we had the current FDA Commissioner. What guidelines, if any, are there for I&I indications and precision approaches? What are some of the challenges and questions that you guys have today for the agencies?
Yeah, I mean, I think that there are guidance, right? And they aren't specific to I&I. There's broad stroke guidance around biomarker development that you can follow. I think what we're seeing, at least at Q32, is a very collaborative FDA on the other side, right? Really looking for ways within cardiorenal and rheumatology to think about these diseases differently, right? Is there a way for FDA to help take the totality of the data you bring in from biomarkers and drive that forward? So we're encouraged by that, but also recognizing that's a long way from getting a drug over the line, right? And getting data that they're going to accept. And so I think we are kind of in our infancy of these discussions with regulators, but I'm pleased to say we're certainly feeling like there's receptivity on the other side. And I think across a lot of divisions at FDA, a desire to move in the direction of oncology, but in a stepwise fashion. And we're at the very early stages of that, I would say.
Would anyone else like to comment on any
Regulatory.
experiences?
Well, I would just say that every indication from clinicians, patients, researchers is that precision approaches in I&I are the future. I have enough faith in the regulators to know that they follow those conversations closely, but there hasn't been any precedent so far, or there's minimal precedent.
Yeah.
So we're all testing it out for the first time, but obviously regulators want to get the therapeutics that patients need and that clinicians want. So I feel like this is a solvable problem.
Good. That's a hopeful note. That's great. I guess on that, too, I think 2 of 3 of you are at least focused on inflammatory bowel disease. And so could you speak a little bit about how you chose your indication to start off and what drove that decision and which disease states you're looking at first?
Yeah, so maybe I can start and then Jason.
Sure.
So 2 reasons for us. One scientific and one personal. So the scientific reason is, as I mentioned earlier, we have a platform based on getting tissue samples from patients and performing single-cell RNA sequencing on those samples to better understand the cellular milieu of disease. And so for an initial indication, we clearly needed to pick something where tissue access was possible. And in the context of that, then we have collaborations where we get tissue samples from colonoscopies, so pinch biopsies of a patient's gut, which we can then look at the cellular milieu. The personal reason is that one of our founders has a child with a serious inflammatory bowel disease. So that founder was very, very motivated to start thinking about new approaches to precision therapeutics in that space. That's what got us off and running.
Yeah, so kind of a similar process for us. I mean, as we mentioned earlier, if you're going to think about a precision approach, what do you want to do? Go find an area where there's an efficacy ceiling. So IBD still, despite a number of different approved therapies being available, a number of different therapies in the pipeline, it's still an area where we struggle to get more than 20+% over placebo in our trials, which is a real challenge. So that was one of the things, that's not the only indication by far. I think one of the other things that, especially as an earlier company, you have to look at indications where if you've got to get to some early data to see, is my approach working, is my therapy working, you need to find some indications where there's earlier hard endpoints. That's a real challenge. Anyone that's in the development business knows that the less opinion and the more measurements that are taken, you feel a bit more confident. There's always still a challenge, but that was a big one for us. And of course, the biology. So you follow the science. That's where on a platform side, we had excellent data, and separately, we also had an excellent set of preclinical data for our lead programs. And so very similar, you line those up and you filter down to the particular indication. For us, we're excited, to progress there and beyond as well, but it's a great starting point.
Yeah, maybe I'll add for Q32, the approach is actually very similar to what's been outlined. So we utilize these C3D staining approaches within biopsies that have been taken across a number of renal and skin disorders, looking at where this co-location with the proteases was taking place. So that was a core driver of indication selection. We had the sort of wealth of riches, right? Because we found it was 80% in a lot of the indications we looked at. So we're targeting ANCA-associated vasculitis, lupus nephritis, IgA neuropathy, and we're also looking at C3G. So we've got these renal basket and the AAV study is the approach that we're taking, and we're moving those forward pretty rapidly. The approach we took was driven by the science, but also I think in the vasculitis side, I just have a personal rationale for it as well, given that my daughter had a vasculitis as a child that occurred a number of times. And so having lived through that and seen it as a parent, the ability to move therapies forward is certainly something that's very motivating for me.
That's great. I think just a comment that I'd make, that I realize that all of you have a lot of interactions with the clinical world, that you need clinical data, you need clinical samples. I think that maybe also that drives a little bit of the science, the availability of the samples and the data out there and these collaborations. And I don't know if anybody wants to speak to that and how that may decide which indications, are developed first.
Huge driver of, I think, as we think of indications, I'm sure you'll both agree, I think the collaborations and discussions we have with clinicians in the field and the KOLs that are out there in these different indications help us think about how we can target those populations, but also help us think about the sort of what's being used as the standard of care and where we could actually fit in the spectrum of the standard of care. Because it's all well and good if you can have an effect, but if it's not going to be something that's utilized, right? If you don't get the endpoints that are going to be necessary to use those drugs, ANCA-associated vasculitis is a good example, right? Steroid sparing being a critical part. And in the absence of steroid sparing, docs don't know how to use the drug. And so as you're building your development, having those relationships with KOLs that are helping to feed that information, and you're making sure you're getting the right answers along the way, so you'll end up with a label that actually supports a drug that's going to be utilized. I think that's a critical part of those relationships.
Another thing is that when we interact with clinicians and we show them our data sets, certain things make their eyes light up. And it's not always the things that we would expect. And why that's important is because if we want to have a shot at developing a new therapeutic in IBD, as we all know, recruitment in those trials is really, really difficult. So we need to be able to also understand what is it in our data sets that's going to get clinicians excited about putting a patient on an experimental therapeutic with a 50-50 chance that that patient's going to get placebo. That is a heavy lift, especially for someone who's really sick. So a big part of interacting with clinicians is actually better understanding our own data, what makes it compelling, and to Jody's point, where what we've got actually fits in the spectrum of therapeutics that's available today.
Yeah, couldn't agree more. At the end of the day, it's the clinicians that end up driving new modalities, new medicines, new practices into the field. And you said something very important, I think, at the beginning of the question, which is for anyone that's looking to bring something innovative in, we have to start with the body of knowledge that exists. While there may be a rare disease in the autoimmunity field that it just seems exactly the place where you may want to take a precision approach by whatever definition. If you don't have a bolus of data sitting there, unfortunately, it lasts a long time. That's why these registries are important. It's why interest in collaboration is important, and that feedback loop, where someone can help you better understand your own data and how exciting or not exciting it is. You want to focus on the right area is very, very critical. So thank you to all the clinicians that look to partner.
I like to hear that.
I'll just add one thing too. I think one of the things we all talk about, like what we can gain from those relationships, but a key part of it is it's the conduit to the patient, right? It's the voice of the patient coming through the KOLs and the clinicians that we're speaking to that help us make sure what we're doing is going to matter to patients, right? At the end of the day, that's the goal, that's the mission, is to bring a drug forward that's actually going to impact patients' quality of life. And it's, you can read a paper and you can make a determination of how your drug's going to fit in, but talking to patients eventually, but also directly through these clinician relationships, I think is a key part of how we hone in on that.
Yeah, and that is something that I should have said when I was talking about autoimmune diseases in general and any inflammatory diseases, that I think steroid sparing is today's holy grail, right? Yeah. And avoiding infection and off-target effects. So if you can do those, that's probably what a lot of the KOLs eyes will light up about. New medications and new indications that will allow us to reduce the steroids, which really we've used way too much of for way too long. They work quickly, and they have horrible deleterious effects long term. So we really need to get away from
Key driver of mortality, right? There's the use of
huge mortality
steroids.
with steroids. So we need to get smarter, and it is exciting.
Totally agree.
I think. And did you have another question? Sorry.
We are actually running low on time, so we could go on forever, but let's go ahead and wrap up this panel. Thank you so much, everyone, for speaking. Thank you, Karen, for co-moderating.
Any last words?
Thank you to the organizers for having us. I think it's a great panel and a great topic we're certainly passionate about.
Yeah, absolutely. This is, I think, this is the area to watch. I think we are moving into a very exciting precision immunology time, and the next 2 to 5 years, I think, is going to be exceptionally exciting. Thank you for organizing.
Yeah, and I'll just say, look, when I was in medical school, I was seeing patients with IBD, and a doctor said to me, you know, this is probably 5 different diseases, but we all just call it ulcerative colitis. And someday, someone's going to figure out that there are actually 5 different diseases here, and we're going to figure out how to treat them. And that's exactly what we're all doing up here.
Exactly. Yeah, this has been a great panel. Thank you very, very much. Thank you, Susan. I've learned a lot.
Thank you.
Thank you. All right.